right? So in this class today we will discuss respiratory energy balance sheet and amphibolic pathway. These two concepts we will discuss. Now, in this respiratory energy balance sheet, we say that one glucose yield 36 ATP molecule. So on complete oxidation of one glucose, 36 ATP molecule will be produced, we say, right? See, energy which is released here from one glucose molecule, we can calculate. We can calculate it is theoretical. It is theoretical and it is based on certain assumptions. The energy released from aerobic respiration on the complete oxidation of the respiratory substrate, it is going to release 36 ATP molecule. So the release of energy from a substrate, it can be calculated. It is based on theoretical information and it is based on certain assumptions. Okay. So what these assumptions we have to learn? Important for 2 marks. So assumptions. What assumptions are there? Listen. We have sequential steps in respiration. Okay, so the steps are sequential and orderly. The steps are sequential and orderly. Means one substrate is converted into another substrate, or another compound, whatever it is. See, we have mainly 3 steps. One is glycolysis. Then the second one is Krebs cycle. Okay. Then the third one is ETS and oxidative phosphorylation. These are the steps. So, in case of respiration, the steps are sequential and orderly. Here, one substrate is converted into another substrate. So, these steps occur one after the other in the sequential order. This is one assumption. These steps occur one after the other in sequential, in orderly manner. This is one assumption. Then the second assumption, what it says, NADH2 is produced in cytoplasm during glycolysis. Okay, so NADH2 which are produced in the cytoplasm from glycolysis. This NADH2 from cytoplasm, it has to be transported to mitochondrial matrix. NADH2 produced in cytoplasm from glycolysis has to be transported. It has to be transported to mitochondrial matrix. Mitochondrial matrix where NADH2 is subjected to oxidative phosphorylation. So second assumption, what it says, NADH2 produced during glycolysis in cytoplasm has to be transported to the mitochondrial matrix where it is undergoing oxidative phosphorylation, and only after oxidative phosphorylation only NADH2 oxidation is going to release. Then the third assumption. So what is the third assumption here? None of the intermediate is used in the synthesis of other organic compounds. Okay, various intermediate compounds are produced. We learned in Krebs cycle. In Krebs cycle, citric acid, isocitric acid, oxalosuccinic acid, alpha-ketoglucaric acid, succinic acid, fumaric acid, malic acid, like that various intermediate compounds are formed. Third assumption, what it says, none of the intermediate compound is used in the synthesis of other organic compound. None of the intermediate compound is used in the synthesis of other organic compound. So the intermediate compounds which are produced during respiratory pathway, they can be utilized in the synthesis of other organic compounds. But what it says, none of the intermediate compound, okay, which is produced in the respiratory pathway is used in the synthesis of other organic compounds. So this is third assumption. Then the fourth assumption, what it says, the most commonly used respiratory substrate is glucose. Glucose is the respiratory substrate. Glucose is the most commonly used respiratory substrate. Okay, see what it says. Only glucose is used as a respiratory substrate. No other substrate is used in the respiratory pathway. Usually glucose is used as a respiratory substrate. Other kind of substrate do not enter respiratory pathway. Other type of substrate, it can be fat or it can be protein, it can be organic acid, whatever it is. Other type of respiratory substrate do not enter the respiratory pathway. So no other substrate is used in the respiratory pathway. Only glucose is used as respiratory pathway. So these are the four assumptions on which theoretically we can calculate the number of the ATP released from a commonly used respiratory substrate, that is glucose. These assumptions which have been made are not valid. They are not valid because all these steps can occur simultaneously, not one after the other. All the steps can occur. Which step? Glycolysis, Krebs cycle, ETS with oxidative phosphorylation. These can occur what we call simultaneously. These assumptions are not valid. Respiratory substrates which can be used, other than the carbohydrate, fats can also be used, proteins can be used, even in some cases like succulent xerophytes, organic acids also can be used. Okay, so these assumptions are not valid. So now we will calculate how many ATP molecules will be produced from a single glucose when it is used as a respiratory substrate on complete oxidation. Actually, the intermediate compounds which are synthesized during Krebs cycle are in turn used in the synthesis of other organic compound. Assumption says no intermediate compound is used in the synthesis of other organic compound, but it is not true. Intermediate compounds are also used in the synthesis of other organic compounds. From glycolysis. From glycolysis. See, step number 1, step number 3. One is phosphorylation, glucose to glucose 6-phosphate. Third is fructose 6-phosphate to fructose 1,6-diphosphate. Okay, here ATP used. We have to mainly discuss about ATP produced, synthesized. Main this. So step number 1, 1 ATP is used. Step number 3, 1 ATP used. This will remember. Okay. Now we have step number, what you call it, 7 and step number 10. Step number 7, step number 10. Step number 7, what is that? 1,3-dipga to 3-pga. Is it or not? 1,3-diphosphoglyceric acid to 3-phosphoglyceric acid. Okay. So there, how many ATP will be produced? 2 ATP will be produced. These ATP are directly produced by which process? Substrate level phosphorylation. Then step number 10, phosphoenol pyruvic acid to pyruvic acid. These steps are remember. Phosphoenol pyruvic acid to pyruvic acid. Step number 10. Right? So here also how many ATP will be produced? 2 ATP will be produced directly by substrate level phosphorylation. Okay. So step number 7, what is that? 1,3-dipga to 3-pga in this step. And step number 10, that is phosphoenol pyruvic acid to pyruvic acid. Is it or not? Right? So here glucose to glucose 6-phosphate and here fructose 6-phosphate to fructose 1,6-diphosphate. So remember this. Now, step number 6. Step number 6. 3-pGAL to 1,3-dipga. Step number 6, you can refer that glycolysis or EMP pathway. 3-pGAL to 1,3-dipga. Check out. Written up? There, 2 NADH2 are produced. There, 2 NADH2 are produced, right? So here, step number 6, 2 NADH2 are produced. Now, each NADH2 will be subjected to oxidation during electron transport system. We learned in the previous class, one NADH2 on its oxidation yield 3 ATP. Right? So now, 1 NADH2, 3 ATP it yield. So how many are there here? 2 NADH2 into 3. Is it or not? I will write here. Down only a point. So here 2 NADH2 into 3 means 6 ATP. So I will write here. So here how much it is? 6 ATP. These 6 ATP are produced after ETS. Is it or not? After electron transport system. After oxidation. Right? Now, from preparatory step, from gateway step, from link reaction, glycolysis over. Okay. See, from glycolysis 6 plus 2 plus 2, 10. 10 produced, 2 used. So total 8. 8. Okay, total 8 after ETS from glycolysis. Number of ATP contributed from glycolysis after electron transport system. That remember. Okay. Now, see this cytosolic NADH2. I told in the previous class, it will undergo shuttle. I told. Okay. Afterwards, once again, I will tell that. Now, from preparatory step, or link reaction or gateway step, whatever you call. So in the preparatory step, here 2 NADH2 are produced. 2 NADH2, that is a oxidative decarboxylation step, where pyruvic acid is converted into acetyl coenzyme. 2 molecules of pyruvic acid, 3-carbon compound, subjected to oxidative decarboxylation, converted into 2 molecules of 2-carbon compound, that is acetyl coenzyme. Right? So yeah, so step number 6, that is 3-pGAL to 1,3-dipGAL. Remember this. And here, what is that? Pyruvic acid to, what is that? Acetyl coenzyme. That is preparatory step. So 2 NADH2 are produced. You know that 1 NADH2 on oxidation yield 3 ATP. So 2 NADH2 are there. So 2 into 3, how many? 6 ATP will be produced here. So your idea 6 ATP from which? From preparatory step. Right? Okay. Now from Krebs cycle, from citric acid cycle, from TCA cycle. Okay. Now step number 4, 6, and 10. Step number 4. What is that step number 4? Isocitric acid to oxalosuccinic acid. Check out that. Isocitric acid to oxalosuccinic acid. Step number 6. Alpha ketoglutarate acid to succinyl coenzyme. There are last malic acid to oxaloacetic acid. Okay. In each step, 2 NADH are produced. So 3 steps to each means how many NADH will be produced? Yeah, 6. 6 NADH will be produced. In this 2, in this 2, in this 2. Right? So how many NADH total? 6 NADH are produced. Okay, 1 NADH oxidation yield 3 ATP. So 6 into 3. How many ATP it can yield? Oxidation of the ETS 18 ATP. Okay, oxidation of this NADH2. Now, step number 8. Succinic acid to fumaric acid, there FADH2 is produced. Succinic acid to fumaric acid, right? So there how many FADH2 are produced? 2 FADH2 are produced, right? So 2 FADH2 are produced. 1 FADH2, how many ATP? 2 ATP. 1 FADH2, 1 FADH2, it yields 2 ATP. Means 2 into 2, how many ATP on the oxidation of FADH2? 4. So you write here 4 ATP. Right here 4 ATP. Okay. Now, in the step number 7, in the step number 7. So what is the step number 7? That is succinyl coenzyme to succinic acid substrate level phosphorylation. There GTP are produced. 2 GTP. GTP is equal to ATP. So step number 7. So there 2 GTP will be produced by SLP. So what we say? GTP is equal to ATP. That means total how many ATP will be produced in the step number 7? 2. 2 ATP. Right? Okay. So now you total it now. You total it. 4, 10, 16, this is 34, 38 to 40. 40 ATP. Now, here how many used? So 40 ATP minus 2 ATP because these are used in the preparatory step for initial reactions of glycolysis. Okay, so now how much now? 38 ATP molecule. 38. 1 glucose, 38 ATP molecule. Now we have to get 36. 36 we have to get. Okay. Now remember, glycolytic NADH2, this one. This glycolytic NADH2 cannot directly enter into what you call oxidative phosphorylation in the mitochondria. Okay. So this glycolytic NADH2 is subjected to what you call a shuttle. It is subjected to shuttle. Here you have one is glycerol phosphate shuttle. Glycerol phosphate shuttle. Glycerol phosphate shuttle. Okay, this can occur in flight muscle cells. Flight muscle cells, in the brain cells, in the nerve cells, it can occur. Glycerol phosphate shuttle. See, in prokaryote, one glucose can yield because respiration reaction occur in the cytoplasm only. Because in prokaryote, no mitochondria. No mitochondria. Okay, so shuttle absent in prokaryote. In prokaryote, one glucose yield 38 ATP. There is no mitochondria. There is no shuttle involved, right? So glycerol phosphate shuttle occur in flight muscle cells, in the nerve cells, or in the brain cells. It occur, right? So when this glycerol phosphate shuttle occur, it is less efficient. It is less efficient. So what happens in this glycerol phosphate shuttle? If NADH2 produced in cytoplasm during glycolysis has to be oxidized, so it makes use of 2 ATP, 2 ATP use. So when glycerol phosphate shuttle occur, for the oxidation of NADH2, okay, during this process, how many ATP used? 2 ATP used. So if 2 ATP is used means 38 ATP minus 2 ATP, how much it comes now? 36 ATP. One glucose, 36 ATP. Remember, this is how we can set 36 ATP. Okay. Now you have another shuttle. This is one type. You have one more type. One more type is malate aspartate shuttle. These are there in higher reference book. Just for information, I am giving you. Okay. So it is malate aspartate shuttle. This is efficient. This shuttle is efficient. Okay. It occurs in what I call the kidney cells. This can occur in kidney cells, all right? So here, no use of ATP here. No use of ATP. No use of ATP means one glucose can yield. See, if NADH2 is oxidized here during this malate aspartate shuttle, ATP is not used. That means one glucose can yield how many? 38 ATP. NADH2 produced in cytoplasm during glycolysis. Okay, either it can undergo like glycerol phosphate shuttle in some cells, or it can undergo malate aspartate shuttle in some cells. Okay, in eukaryotes, higher eukaryotes are that. So there, if this shuttle occur, no use of ATP during the oxidation of cytosolic NADH2. That means as it is one glucose can yield 38 ATP molecule. So 38 is also correct, 36 also correct, but depends on the shuttle, the cytosolic NADH2 undergoes. Which shuttle it undergoes, whether it is a glycerol phosphate shuttle or malate aspartate shuttle. So this you remember. Okay, simple what we can say in simple. Okay, in simple, the NADH2 produced during glycolysis in cytoplasm. Okay, these two NADH2 are produced during glycolysis in cytoplasm to transport to mitochondria. To transport to mitochondria, where it undergo electron transport system, where it undergo oxidation process. Okay, so for this, to transport one NADH2, one ATP is used. This is simple meaning. Actually, shuttle occurs. Simple meaning I am giving. Okay, to transfer NADH2 produced during glycolysis in cytoplasm, these NADH2 have to be transported to mitochondria, where they are subjected to electron transport system and oxidation process occur. Okay, so for transport of one NADH2 from cytoplasm to mitochondria, one ATP is used. How many NADH2 are there? Two NADH2 are there. So it make use of how many ATP? Two ATP. So what we will write? 38 ATP minus 2 ATP if they are used, then how much you will get? 36 ATP from 1 glucose. But this is simple meaning. Actually, there will be operation of a shuttle by which cytosolic NADH2 is oxidized. Then either it can yield 36 ATP or it can yield 38 ATP. This is the extra information you try to remember. Otherwise, just you have to remember the simple meaning. Okay, why these NADH2 are transported from the cytoplasm to the what you call mitochondria. So they have to be subjected to oxidation. So their expenditure of ATP will be there. That's why net it will be 1 glucose 36 ATP. Right? So this is about the respiratory energy balance sheet. Okay, so try to remember this. Very important. Listen. In the previous class, I told calculation of 6 carbon dioxide releases. Where in which steps carbon dioxide releases I have told. Okay. Now, in respiration, how many oxygen will be used? 6 oxygen will be used as per the respiratory reaction. Where exactly oxygen is used? In glycolysis, oxygen is not used. In Krebs cycle, there is no direct utilization of oxygen. So oxygen is used mainly in terminal oxidation process. Terminal oxidation process. Okay. Remember, from glycolysis, 2 NADH2 are produced. Okay. See, we wrote half molecule of oxygen. It receives 2 proton and it receives electron from cytochrome A3, 2 electron from cytochrome A3 and get converted into metabolic water. So here is the step where oxygen is used, right? So when one NADH2 is oxidized, half oxygen molecule is used. Half oxygen molecule is used, is it or not? NADH2 are oxidized. How many oxygen will be used? One oxygen. Remember this. So glycolysis yield 2 NADH2. So here one oxygen is used during the terminal oxidation step. Okay. Similarly, in the preparatory step or gateway step or leak reaction, how many NADH2 will be produced? 2 NADH2. So how many oxygen can be used? One oxygen. Is it or not? Right? Then the next Krebs cycle you take. Krebs cycle, total 6 NADH2 from 2 terms. Okay. If 6 NADH2 are used, then how many oxygen here? 3 oxygen. And how many FADH2? 2 FADH2. So how many oxygen here? 1 oxygen. Right? 3 plus 1, 4 plus 5 plus 6. So 6 oxygen molecules will be used during terminal oxidation when ETS occur on the inner membrane of the mitochondria in cristae. Correct? 6 oxygen molecule. What the calculation? Now similarly, water availability. Water. Usually, how many water molecules released on the product side? Product side 6 CO2 plus 6 H2O, 6 water molecule. Now, that water molecule calculation will give. Now, see here. When one molecule of NADH2 undergo oxidation, how many water molecule it is going to form? One. Is it or not? One NADH2 or one FADH2 on oxidation yield one water molecule. So total how many NADH2 we have? 2 from glycolysis, 2 from preparatory or Link reaction, and 6 from Krebs cycle. Total how many? 10 NADH2. Okay. So these 10 NADH2, when they are subjected to oxidation after terminal oxidation, right? So how many water molecule they can form? 10 water molecule they can form. 10 water molecule. Right? How many FADH2 are there? 2 FADH2. So after their terminal oxidation, how many water molecule they can form? 2 water molecule they can form. 2 water molecule. Right? Now, recall. Krebs cycle. Krebs cycle. Step number 2. Krebs cycle. Step number 2. 2 water molecule will be released. Citric acid to cis-aconitic acid. 2 water molecule release here. Right? 2 water molecule release. Step number 2 of Krebs cycle. Krebs cycle. 2 water molecule release. Right? Then you take glycolysis. Glycolysis. Step number 9. Step number 9. 2 phosphoglyceric acid to phosphoenol pyruvic acid. Step number 9. Is there a 2 phosphoglyceric acid to phosphoenol pyruvic acid is catalyzed by analysis enzyme. There 2 water molecules released. Released or not? Okay. So step number 9. 2 water molecules released. Okay, now you see here. Total how many water molecules produced? How many water molecules produced? 16 water molecules produced. These are all produced. These are all produced. Now, water molecule used calculation. Water molecule used. Okay. Now, you take Krebs cycle. Krebs cycle. 1.
1053. 105. Yeah, what is it? 1053. Outside that. How much back in body? No. No idea.
See, in Krebs cycle, step number 1, step number 3, step number 7, step number 9. In one term, 4 water molecule used. After 2 term, how many water molecule used? 8 water molecule used. Step number 1, 3, 7, and 9. One term, 4 used. After 2 term, how many? 8 water molecule used. Okay. Now, in the reference book, they have given glycolysis. Glycolysis. Step number 6. Step number 6. That is 3-PGAL to 1,3-dipGA, where phosphoric acid is used. Okay, where NADH2 is produced now. Step number 6 in glycolysis, NADH2 is produced, phosphoric acid is added. Okay, so there 2 water molecules used. It is there in reference books. Okay, so step number 6, 3-PGAL to 1,3-dipGA, right? So there it is a dehydrogenation reaction or oxidation reaction where NADH2 is produced. There 2 water molecules are used. So total how many used here? Total how many used? 10 water molecules used. How many produced? 16 water molecule produced and 10 water molecule used. Okay, so net how many water molecule here? 6 water molecules. So that's why on the product side you have 6 H2O along with 6 carbon dioxide. So where water is produced, where water is used, where oxygen is used, where carbon dioxide is produced, all the calculations over. Right? So this is all about what you call electron transport system, oxidative phosphorylation, Krebs cycle, everything I have completed. Right? So got the idea now where water molecules produced, where water molecule used, where carbon dioxide is released. I have given preparatory step 2. Okay, step number 5, step number 6 in Krebs cycle. There 2. So 4 in Krebs cycle, 2 in preparatory, total 6 carbon dioxide. Right? So these things we have to remember. Okay, next now we have to learn the differences between aerobic respiration and fermentation. Differences between aerobic respiration and fermentation. Aerobic respiration. This is fermentation. First, it occurs in all higher organisms. Aerobic respiration, it occurs in all higher organisms. It can be plant, it can be animal, human being, okay, whatever it is. So it occurs in all higher organisms. Higher organisms. This it occurs in certain bacteria, certain fungi, right? So it occurs in certain bacteria and fungi. Okay, and I told skeletal muscle cells when they are involved in heavy exercise, heavy running, the muscle cells of the animal, even human being, they perform fermentation. When the muscle cells do not get adequate oxygen, then temporarily they can perform fermentation. So in that case also you can see fermentation. But otherwise usually it occurs in certain bacteria, certain fungi. So prokaryotic bacteria, eukaryotic certain fungi. There you can see this fermentation. Here in aerobic respiration, substrate, respiratory substrate, that is glucose, is completely oxidized. Completely oxidized. Respiratory substrate is completely oxidized. Here it is the glucose. Respiratory substrate is incompletely oxidized. Incompletely oxidized. Aerobic respiration, it occurs in presence of oxygen. In presence of oxygen. This in absence of oxygen. This process occurs in absence of oxygen. Here, several intracellular enzymes. It is an intracellular process. It is an intracellular process. Usually fermentation is extracellular process. Extracellular process. Here, several other enzymes are involved. Other enzymes are involved, especially in Krebs cycle, in EDS, all that other enzymes are involved. Here, which enzyme is involved? Zymase enzyme complex. Zymase enzyme complex is involved. Zymase enzyme complex. Usually, end products are here. Which will be the end products? Usually, carbon dioxide and water. So here, the end products and the products are usually ethyl alcohol. Ethyl alcohol, right? If it is alcoholic fermentation. If it is a lactic acid fermentation, what will be produced? Lactic acid. Lactic acid can be produced. In lactic acid fermentation, no liberation of carbon dioxide. In alcoholic fermentation, liberation of carbon dioxide. Ethyl alcohol with liberation of carbon dioxide. Okay. Then here, the energy released. How many molecules of ATP? 36 ATP molecules are released. Is it or not? One glucose can yield 36. 36 also correct, 38 also correct. Right? So 36 ATP molecules released. And here, one glucose, how much energy it releases? Only 2 ATP molecules produced. Here 36 ATP, there are only 2 ATP molecules produced. Okay? So here, NADH2 undergo vigorous oxidation. Vigorously it undergo oxidation. Rapidly it undergo oxidation to yield ATP. Here, NADH2 will be oxidized slowly. NADH2 will be oxidized slowly. Here it is rapidly oxidized. Here it is slowly oxidized. Okay. So these are some simple differences between aerobic respiration and fermentation. So that you try to remember. Okay. So these differences you try to remember. Very important for 2 marks. For this one, finish up. See, aerobic respiration, here mitochondria is involved. Here, no mitochondria. Right? Aerobic respiration, what it involves mainly here steps. It involves usually glycolysis, then Krebs cycle, and ETS, along with oxidative phosphorylation. Here it involves only glycolysis and fermentation. Glycolysis and fermentation. These are the sequential steps. Okay, that is glycolysis, Krebs cycle, and ETS, along with oxidative phosphorylation. Here, only glycolysis will be there with fermentation. So this is another difference. If you want, you can write the reaction also. In aerobic respiration, what will write? C6H12O6 plus 6O2, 6 carbon dioxide plus 6H2O plus 36 ATP. This is aerobic respiration reaction. If you take this anaerobic fermentation, C6H12O6, under anaerobic condition, it is giving rise to 2 molecules of ethyl alcohol plus 2 carbon dioxide and only 2 ATP. This is the chemical equation, chemical reaction difference. Okay, moving on to the next one now. Amphibolic pathway. Amphibolic pathway. Listen. Usually what we say, respiration is a catabolic process. Usually what we say respiration is a catabolic process. The most commonly used respiratory substrate, that is glucose, is broken down into carbon dioxide and water. Okay. So when this glucose is used, it enters the respiration from first step. From first step, that is from glycolysis, glucose 6-phosphate, fructose 6-phosphate, like that. Okay. So this is one of the most commonly used respiratory substrate. So this complex sugar is broken down into simple sugar. That simple sugar can enter right from the first step of the respiration, that is glycolysis, whatever it is. Now, in some cases, other respiratory substrates are also used. Say, fats can be used, fats or lipids can be used. These fats or lipids, first they should be subjected to oxidation. That is, they should be broken down into simple molecules, right? So these fats or lipids, they are subjected to beta oxidation. Okay, so they will be converted into simple molecules such as fatty acids and glycerol. Fatty acid and glycerol. Directly fats cannot enter respiratory pathway. First, they should be converted into simple molecules. They should be broken down into simple molecules. That is fatty acid and glycerol. Now, this fatty acid cannot enter the respiration from first step. Fatty acid cannot enter the respiratory pathway from first step. Suppose if fatty acid is used as a respiratory substrate, it is used as a respiratory substrate, this fatty acid is converted into acetyl coenzyme. So directly, fatty acid is converted into acetyl coenzyme. This acetyl coenzyme then take part in Krebs cycle. That means if fatty acid is the substrate, glycolytic steps not required. Directly, it is converted into acetyl coenzyme. Okay. Suppose if glycerol is the substrate, if glycerol is the respiratory substrate, it is converted into 3-PGAL, 3-phosphoglyceraldehyde. 3-phosphoglyceraldehyde, as usual, you will get after what I call fifth step, you will get. After fifth step, you will get 3-PGAL. Okay. So that 3-PGAL, remaining 5 steps of glycolysis as usual will be there. 3-PGAL to 1,3-dipGA, 3-PGA, 2-PGA, phosphoenol pyruvic acid, pyruvic acid. Pyruvic acid will undergo preparatory step, acetyl coenzyme, and then Krebs cycle. So this 3-PGAL is produced from. Okay, this is in case of fats or lipids. Suppose if proteins are there, at the respiratory substrate. So these proteins, see, fats will be broken down into simple fatty acid and glycerol by the action of lipase enzyme. Is it or not? So usually, if sucrose is there, sucrose is C12H22O11. This will undergo hydrolysis. When it undergo hydrolysis, you will get glucose. Okay, so this is broken down. Usually, which enzyme will you say? Invertase. This is invertase. Okay, sucrose is broken down into glucose and fructose. Glucose and fructose into invertase. So here lipase. So proteins can be converted into simple monomeric units. These are amino acids. Okay, the enzymes which will break proteins, protease enzyme. Right. So these amino acids will enter the respiratory pathway. They don't enter from the beginning. Either fats or protein, they don't enter from first beginning. Beginning means glucose 6-phosphate, fructose 6-phosphate, what we learned in glycolysis. Okay, so amino acid. Some amino acids are produced, during the breakdown of the protein into simple amino acid. Say if glycine is produced, okay, from protein, this glycine is converted into acetyl coenzyme, then it takes part in Krebs cycle. Glycine, this amino acid. Suppose if the amino acid is alanine, If the amino acid is alanine, it is converted into pyruvic acid. Pyruvic acid is actually the end product of glycolysis. End product of glycolysis. So alanine is there means it is converted into pyruvic acid, and from pyruvic acid, as it is enter respiratory pathway. Okay, so depending upon the what I call respiratory substrates used here, they enter the respiratory pathway at different stages. So initially it involves catabolism where breakdown of the complex organic compounds will be there. Carbohydrates, fats, proteins. These are the what we call the respiratory substrates. These are different respiratory substrates. These substrates can be used during respiration. So first, if the carbohydrates are there, these carbohydrates are broken down into simple sugars. Simple sugar. That simple sugar is mainly glucose. Simple sugar is mainly glucose. This glucose is converted into glucose 6-phosphate. Glucose 6-phosphate to what will get? Fructose 1,6-diphosphate. Complete glycolysis I am not writing as per NCERT. Okay, this fructose 6-phosphate splits into one that is DHAP, dihydroxyacetone 3-phosphate, and another one what is that? 3-PDAL. This 3-PDAL, okay, it is converted into what? Pyruvic acid. Is it or not? At the end, it is converted into pyruvic acid. This pyruvic acid, after respiratory step, it is converted into acetyl coenzyme. This acetyl coenzyme, where it enter? Krebs cycle. When Krebs cycle occurs, completely it is decarboxylated into carbon dioxide and water. So normal if glucose or sugar is used as respiratory substrate. Now fats can also be used. So these fats are converted into fatty acids and glycerol. This fatty acid usually it is converted into what is that? Acetyl coenzyme. Suppose if this glycerol is used, then this glycerol can be converted into water. This is PGAL. PGAL and DHAP, these are which forms? Isomeric forms. Is it or not? They are usually isomeric forms. So glycerol, if there means it is converted into 3-PGAL. Okay. Now say here, proteins. Proteins will be usually broken down by the action of protease into amino acids. Okay, these amino acids enter the respiratory pathway. If alanine is there, it is converted into pyruvic acid. From there it takes form. Suppose if lysine is there, it is converted into acetyl. Okay, so this is the figure interrelationship of major pathway showing interrelationship of major pathway showing respiration mediated breakdown of organic molecules into carbon dioxide and water. Carbon dioxide and water. So this schematic figure, interrelationship of major pathway, that is major what you call metabolic pathway, whatever you write, major pathway or metabolic pathway showing respiration mediated. Through respiration only, these are broken down. Okay, finally into whatever the respiratory substrate is used, wherever let it enter into respiration process. Ultimately, either you get pyruvic acid or acetyl coenzyme, then there will be operation of Krebs cycle, and acetyl coenzyme is completely decarboxylated into carbon dioxide or carbon dioxide and water. Right? So this you try to remember, very important. This Krebs cycle is called an antibiotic pathway. Krebs cycle is known as antibiotic pathway. Why it is called an antibiotic pathway, I will take up in the next class. So in the next class, I will complete this chapter. One concept is the respiratory portion. So that's the this I will complete. Right? And then you will move on to the last chapter of your syllabus. Growth regulators. Okay, that require around 3 periods. You will wind up your syllabus.